Bismillah Rahman Rahim 9700 June 23 question paper 22 AS level biology, and this is the first video. Question number 1. The sinoatrial node and the atrioventricular node AVN have an important role in the control of the cardiac cycle. The timing of the atrial and ventricular systole. Systole is the contraction and diastole. It's not systole, sorry, it's systole and diastole. So systole is the contraction and diastole is the relaxation. So this is contraction and this is relaxation. Must be controlled so the blood passes through the heart efficiently. Figure 1.1 is a summary of blood flow through the right side of the heart during one cardiac cycle. 3 boxes in figure 1.1 are not complete. Complete boxes 3, 5 and 7 in figure 1.1 using only the terms systole and diastole. Blood arrives at the heart in the superior vena cava. So this is the superior vena cava and this is the inferior vena cava. And blood passes into the right atrium. Then blood will pass. Now what is going to happen? Then it's called atrial systole. So atria is going to contract. So atrial systole. Then ventricular diastole. And then blood is forced into the ventricle. So the blood enters the ventricle. Then what is going to have? Atrial diastole. Atria is going to relax. As I told you, diastole is relaxed. So diastole is relaxed. DR. Make mnemonics to remember things. Then when the atria relaxes, then this ventricle is going to contract. So then this is systole. Systole is contraction. SC, Supreme Court. Systole is contraction. Make some mnemonics to remember things. Blood leaves the right ventricle to enter the pulmonary arteries. Now what is going to happen when the blood goes into then 7 atria is going to still be in a relaxing state. So diastole and ventricular. Now the ventricle is going to relax. Why? Because now the blood leaves the right ventricle to enter the pulmonary arteries. So now the ventricle is going to relax, and that is going to be ventricular diastole. So systole, diastole, diastole, systole, and then diastole, diastole. Because the atria will of course relax, the ventricles will also relax. Part B of the question: impulses sent out by the SAN pass to the AVN where there is a short delay. With reference to figure 1.1, explain why it is important for the control of the cardiac cycle that there is a short delay at the AVN after impulses have been sent out by the SAN. Atria and ventricle do not contract at the same time. Ventricles contract after the atria. So the atria contract first and then the ventricles contract. Now, so this allows the atria to empty, or you can say allows the ventricles to fill up. So all the blood must be pumped into the ventricles, which should then contract after that once it's all full. So it was a little difficult. I know main thing was you wouldn't know how to word it. Otherwise it was an easy question. So atria and ventricle do not contract at the same time. Ventricles contract after the atria, and this delay helps in allowing the atria to empty, or it allows the ventricles to fill. Now coming to C part of the question. Changes in blood pressure occur in the heart during the cardiac cycle. That's fine. These changes cause the opening and closing of the bicuspid and tricuspid valves, which are also called the atrioventricular valves, and the aortic and the pulmonary valves. Explain how blood pressure changes cause the opening of the tricuspid valve, how blood pressure changes cause the opening of the pulmonary valve, and how blood pressure changes help the flow of blood through the heart. So everything is how the blood pressure changes. How the blood pressure changes cause the opening of the tricuspid valve. And so all these in context of that you've got to be talking about. So if you look at all of them sort of separately, opening of the tricuspid valve, pressure in the right ventricles becomes lower than the pressure in the right atrium. Why would the tricuspid valve open? So you've got to understand is that the tricuspid valve is between the right atrium and the right ventricle. So the right side has the tricuspid valve. So it's got 3 flaps. So why would it open? Because pressure in the right ventricle, becomes less than the pressure in the right ventricle. Just like when you're entering a room and the door is closed, so you push the door, so on one side the pressure is more. You push the door and the door opens onto the other side. Like you are outside the room and you push the door, so the door is going to open into the room, and then you're going to enter the room. So pressure in the right ventricles become lower than the pressure in the right atrium. So pressure in the right ventricle is lesser than the pressure. So here the pressure is more, and that pushes these valves open. Why would the semilunar valves be open? Now, why would these semilunar valves open, which are going into the pulmonary artery, and the pulmonary artery carries deoxygenated blood to the lungs? So pressure in the right ventricle becomes more, and then this pushes these open, and then the blood enters the pulmonary artery. So pressure in the right ventricle has to be more, higher than the pressure in the pulmonary artery. So this pressure has to be more than the pressure in the pulmonary artery. And the last point is help blood flow through the heart. Now, one-way flow of blood directs blood in the correct direction. So the valves are preventing the backflow of blood. And blood flows from a higher pressure to a lower pressure. And blood pressure changes result in coordination. Semilunar valve is open when tricuspid valve is shut. So all these valves work in a certain sequence. When this tricuspid valve is closed, then of course the semilunar valve will open and blood will enter the pulmonary artery. So you have to understand it as well as you must know how to write it. You might understand it, but you are unable to write it. So that is a problem, with a dilemma with a large number of students, that they understand it but they don't know how to word the answer. So the answer to the opening of the tricuspid valve is pressure in the right ventricle is lower than the pressure in the right atrium. Right? So I'm giving you the answer to this so that you can see which one is talking about which one. For the second one causes the opening of the pulmonary valve, pressure in the right ventricle is higher than pressure in the pulmonary artery. So the help the flow of blood through the heart, there are three marking scheme points. One-way flow of blood flows from higher pressure to lower pressure, and blood pressure changes aid the coordination of the cardiac cycle. And of course that results in semilunar valve is open when tricuspid valve is shut. Figure 2.1 is a photomicrograph of a longitudinal section through a root tip. Two different regions are visible, the root apical meristem and the root cap. Cells in the root cap synthesize a gel-like sticky secretion known as mucilage, which is important in reducing friction between soil and the growing root. It is composed mainly of polysaccharides and also contains some amino acids and enzymes. Root apical meristem cells, and then we have the root cap cells. So we have this area, and then we have this whole thing is about 1 millimeter, because you can see this measurement here. And then there is the root cap cells, and then if you look at the root apical meristem cells. Describe three differences visible in figure 2.1 between the root apical meristem cells and the root cap cells. Now let's look at them once again. Now if you look at the root apical meristem cells, they're mostly in mitosis, while these cells are larger and they have a larger nucleus. So the root apical meristem cells, if you look at them, they are cells in mitosis or different stages of the cell cycle, while the root cap cells are no cells in mitosis. All cells are in interphase. Then here some cells are in prophase, metaphase, and telophase, while here we don't say any such thing. Then generally the root apical meristem cells are smaller in size, and these are generally larger in size or more cytoplasm. Then less cytoplasm, more cytoplasm. Then you could have larger nucleus in proportion to size of the cell, smaller nucleus in proportion to the size of the cell. Then nucleoli prominent, nucleoli not prominent. Then generally all cells cuboidal, and this is of course variety of shapes, irregular or you can say circular even. And arrangement of cells in rows and loosely packed, more intercellular spaces. More cells have nuclei, some have no nucleus. So a lot of things which you could have said in comparison, and a very easy question for 3 marks. So when you look at the comparison between the root apical meristem cells and the root cap cells, the first point that I'm giving you is some cells in mitosis, no cells in mitosis, then smaller size, larger size, then larger nucleus in proportion to size of the cell, then smaller nucleus in proportion to size of the cell, then nucleolus prominent, nucleolus not prominent, all same shape, variety of shapes, cells arranged in rows, and irregularly, cells are loosely packed or irregularly arranged. So lots of points, and you have to only give me 3 of these, so you were all able to do on these questions. I'm sure it wasn't very difficult. Then coming to the B part of the question, mucilage acts as a glue to bind tiny soil particles together, forming small clumps. So mucilage acts as a glue so that the tiny soil particles together, forming small clumps close to the root. These small clumps help to maintain the soil water around the root tip and prevent the loss of water. With reference to the cohesive and adhesive properties of water, suggest and explain how the formation of small clumps of soil help to maintain the soil water around the root tip. So this is the root tip, and then these clumps form around it because of the mucilage, which acts as a glue to bind the soil particles together. So basically, when we're going to talk about cohesion and adhesion, just describe it, just define it. So water molecules form hydrogen bonds. 1 mark. Cohesion between water molecules. Then cohesion to water molecules adhering to the soil particles. Then adhesion of water molecules to the soil surface. Then clumps provide areas in which water collects. So any 3 out of these, and you would have got your 3 out of 3. Water molecules form hydrogen bonds. Second, cohesion between water molecules. Number 3, cohesion to water molecules adhering to soil particles. Then number 4, adhesion of water molecules to the soil surface. And then clumps provide areas in which water collects. When part C of the question, enzymes present in mucilage catalyzes the breakdown of organic compounds in the soil. This increases the presence of mineral ions in the soil. State the term used to describe enzymes that act outside the cell that synthesize them. Extracellular enzymes. So that was easy. The ones which work outside are called extracellular enzymes. Then part D, the polysaccharides and amino acids present in the mucilage are a source of nutrients for the microorganisms that live in the area surrounding the root. Some of these microorganisms can break down soil compounds to release mineral ions. What is meant by a polysaccharide? Large number of monosaccharides joined by glycosidic bonds, repeating monomers. So repeating monomers, large number of monosaccharides joined by glycosidic bonds. So that is the easiest definition of a polysaccharide. Then coming to the part 2 of the question, which says the soil microorganisms use amino acids to synthesize proteins. All the 20 different amino acids that are present in proteins have the same general structure. Draw the general structure of an amino acid in space provided and use this drawing to explain how it is possible to have many different amino acids. So basically you've got to draw the amino group, and you have to draw the COH group, and you have to show to the left and the right, and the R group is variable, so different amino acids have different R groups. So basically this is the diagram that is expected of you. This would have got you your 1 mark, and the fact that different amino acids have different R groups, or you could have different side chains, any of that, and you could have got your 2 out of 2 for this part of the question. To the last part of the question, mineral ions are usually present in the soil in very low concentration. The action of mucilage enzymes and soil microorganisms can help to increase the presence of mineral ions. Root hair cells are specialized for the uptake of these mineral ions and for the absorption of water from the soil. Suggest and explain how the presence of mineral ions in the root hair cell can increase the absorption of water by the root hair cell. So basically they are saying that inside, if you have too much of nitrate ions and too much of potassium ions and too much of calcium ions, any mineral ions, magnesium ions, anything. So if you have a lot of these, suggest and explain how the presence of mineral ions in the root hair cell can increase the absorption of water. Why would it increase the absorption of water by the root hair cells? So basically what is going to happen? The mineral ions are solutes and they dissolve in the water. So they're going to decrease the water potential. So inside here, there is going to be a decrease in the water potential. Now there is a steeper water potential gradient because here we've got water outside. Why should water go in by osmosis? Because there has to be a lower water potential here. Inside the cell there has to be a lower water potential, and outside there has to be a higher water potential. So increases or steeper water potential gradient, and water follows osmotically. Osmotically, water enters by osmosis. So I hope everybody is clear about this, what they were expecting you to answer and how you were going to answer this. So mineral ions dissolve in water, lower water potential within the cell, increases the water potential gradient, water enters by osmosis. Question number 3, cells of the immune system respond to the presence of non-self antigens. If your cell has this antigen on it, then anything which resembles this is self. But if another virus enters your body with this sort of an antigen on its surface, then this is a non-self antigen. So your body cells recognize your own cells and don't make antibodies against it. But if a virus or a bacteria or a fungus enters, now immediately the lymphocytes know that this is not my own body cell. So I've got to destroy it. There are of course different methods by which we destroy it. So outline the features of the non-self antigens. Number 1, they're usually proteins or glycoproteins. Number 2, they stimulate an immune response. Then they are recognized by or activates the immune system cells like the phagocytes or the B and T lymphocytes. Then they cause production of antibodies, and the plasma cells will release the antibodies. Then the surface pathogen or anything, bacterial cell wall, where is the location of the non-self antigen? Whole cells are non-self antigens. Well, you can't say that. That's not very correct. So surface of the pathogens have antigens on it, and they could also be they are very high molecular weight, and they can be a toxin produced by a pathogen as well. So stimulates an immune response. Antigens are usually proteins or glycoproteins. They stimulate B and the T lymphocytes, and then the B lymphocytes, plasma B cells, make the antibodies, and the surface of the pathogens have the antigens. Now coming to the next part of the question, 4 different types of cells of the immune system are shown in table 3.1. Complete table 3.1. Use a tick if the description applies to the named cell of the immune system. Use a cross if the description does not apply. Able to go through the number of cell cycles, clonal expansion. Read it very carefully. Able to go through a number of cell cycles, clonal expansion. Who is going to go through these? The B lymphocyte, not the plasma cells, the T helper cells, and the T killer cells. You see, once the clonal expansion, then some of them become the B lymphocytes. Then the second point is, main role is to secrete cytokine during an immune response. This one doesn't, this one doesn't. T helpers do, and this one doesn't. Then present during a primary immune response to a virus. B lymphocytes are present, plasma cells are present, T helper cells are present, and T killer cells are present. So you had to get this 1 mark, this whole line correct. So this whole line had to be correct. Then this one had to be correct. Then this, and then this. So you have to get your 4 marks in which the entire line was correct. Then the cell can be divided into different parts. The cell cycle can be divided into different parts. Complete sentence A, B, and C to provide more information about the mitotic cell cycle. The part of the cell cycle that occurs immediately after mitosis is completed. I think there's something missing here. The part of the cell cycle that occurs immediately after mitosis, is cytokinesis. The part of the cell cycle in which the S phase occurs, interphase. The main event that occurs during the S phase is DNA replication. This was easy. That completes this video about the first 3 questions. And the next 3 questions will be solved in the next video. And best of luck and thank you very much. Please do subscribe and thank you for appreciating and liking the videos. Thank you very much.